So thank you so much, everybody, for joining us tonight to learn a little bit more about the EDELIFE clinical trial and the potential treatment for XLHED. My name is Veronica Minard, and I'm the Director of Marketing and Communications for the National Foundation for Ectodermal Dysplasias. And our Executive Director, Mary Fate, was planning to host tonight's webinar, but she had an important family event that corresponded with the timing. And so unfortunately, she couldn't be here. She's pretty bummed that she can't be here with us live. However, she did pre-record some messages for you all, so that'll be a part of our presentation. And I, typically kind of work behind the scenes on everything, but I'm really happy to be here with you all tonight. So thanks for being here. We are asking everybody during the presentation to keep your microphones muted, and we will certainly have question and answer time after the presentation is over. Feel free to turn your video on if you want. If you don't want, that's totally whatever you're comfortable with. That's fine too. We do have a couple of staff members on tonight, so I was going to just ask them to say hi really quickly so you can know, if you haven't met them before, who they are and what their role is with the NFED. So Becky, do you want to start?
Sure, I'm Becky Abbott, and I'm the Director of Research and Treatment Advocacy. I'm also a carrier of XLHED, and my oldest son, Aiden, is affected by XLHED. So thank you so much for everybody being here tonight.
Becky. And Kelly.
Hi, everyone. It's good to see you and be on here with you this evening. My name is Kelly Atchison. I am the Director of Community Programs and Outreach, I think, but I never remember my title. I do a little bit of everything, but the most or best thing about my job is I get to talk to all of you when you call in or email in with questions and provide information to you. So I've been around a long time, so many of you on the call I may have met already, but if not, it's glad to meet you, and I hope to get to meet you in person this summer at Family Conference. Shameless plug, sorry.
I'm glad you did it.
Thank you.
Thanks, you guys. So for tonight, we are going to provide an overview of the EDELIFE clinical trial. We're going to explore the science behind it and behind XLHD and the potential treatment. We're going to learn who might qualify to participate. And of course, we're going to provide with information on how you or a loved one can learn more or get involved and participate with the trial. Most importantly, we really want to allow time at the end for any of your questions and provide answers, because we do have some folks who are experts and experienced with the study here so that they can provide those answers for you tonight. So feel free when we're through the presentation to chime in, unmute yourself, or if you're more comfortable typing in the chat, we'll check that as well. And be sure to try and get all of your questions answered, excuse me. And so we do have some really awesome speakers joining us tonight. As I noted, our Executive Director, Mary Fate, couldn't be here in person, but you will be seeing from her in video. Dr. Kathy Grange is here with us tonight. She is a clinical geneticist at Washington University in St. Louis, Missouri, and that is currently the first and only trial site in the United States for the EDELIFE clinical trial. She's also the principal investigator for the trial at this location. And Dr. Grange is also a member of the NFED's Scientific Advisory Council, and we're very grateful for her leadership and for her sharing her expertise with us here tonight. We have Laura Reiser on with us, and she is an XLHED carrier and also received the potential treatment for her son Bennett back in 2020. And she traveled all the way to Germany during a pandemic to do that. So you'll get to hear a little bit about her story, which is pretty great. And we may have another mom joining us. I don't think that Emily's on, but
she's texting me right now. She's trying to get on.
Okay.
So I'll see if I can help her figure it out.
Okay. let me know if we need to help in
Okay.
any way. So hopefully Emily Nelson will also be with us. She, I think a few months before Laura went over to Germany, had received the treatment, the potential treatment as well in Germany. So we're really excited that both of these courageous moms are joining us and are willing to share their stories and their perspectives on participating in the trial. So I'm going to jump right in with the overview. Oh, and I forgot to advance my slides. Here's a picture of Emily and her family, and Laura and her family. And we're going to jump right in and get started hearing from Mary.
Good evening. I'm Mary Fate. I'm the Executive Director for the National.
It did this earlier. It paused a little bit.
Foundation for Ectodermal Dysplasias, and I want to welcome you to this evening's webinar, and thank you for joining us. I'm really disappointed that I'm not live on the webinar, but my grandson had a special event this evening, and it's something that I can't miss, so I thank you for your understanding. So we have lots of good information for you about EDELIFE. So what exactly is EDELIFE? It's a potential treatment for boys affected by X-linked HED, X-linked hypohidrotic ectodermal dysplasias. And this treatment is a novel approach to a therapy. It is the recombinant protein is injected in utero, so meaning in the uterus of a pregnant mom. This therapy is groundbreaking and really exciting, the first ever potential therapy for X-linked HED. So I want to share just a little information with you. It's been a long time coming, and oftentimes research happens so much more slowly than we'd like to see it. So there's a story behind this treatment, and I want to share that with you. So the NFED has long supported research, and we're very fortunate because our executive director and founder, Mary Kay Richter, was a visionary, and she knew that if the NFED did not fund research for the ectodermal dysplasias, likely it would never be funded. So a little history about this story. So in 1987, a long time ago, NFED funded Dr. John Zonana and his colleagues to map the X-linked HED gene. And in 1996, they were finally successful in identifying the gene that causes X-linked hypohidrotic ectodermal dysplasias. And this is critical to developing any types of treatment. So it was a huge success. So continuing and jumping forward to 2002, the NFED funded Dr. Maggie Cassell to study the dog model of X-linked hypohidrotic ectodermal dysplasias. And this was critical in understanding the syndrome and how it manifests itself. In 2004, the NFED funded Dr. Olivia Gade and Dr. Pascal Schneider from Switzerland, and they successfully developed a recombinant protein that potentially could replace the missing protein in those affected by X-linked HED. So they treated mice with this protein and were very successful in having those mice develop sweat glands and hair. So it's pretty fascinating. Actually, I was fortunate enough to visit their lab and see these mice, and it was just amazing. So in 2007, again, the NFED funded Dr. Maggie Cassell to successfully use that protein to treat dogs that were naturally affected by X-linked HED. And the dogs showed significant improvement in dentition and sweat. And that's just phenomenal. In 2008, we funded Dr. Cassell to continue her work to study this protein in dogs. And in 2010, the NFED developed an international registry so that we could more carefully study and understand the natural progression of the syndrome. So in 2010 and 2011, you may remember the NFED partnered with EDELIFE to study using this protein in humans. And so the first study was a safety and efficacy study where the protein was injected in adults to make sure that it was safe. And we found out it was safe. It didn't cause any harm. And so in 2013 to 2015, trials began for newborns. And this protein was injected within 14 days after the birth of a baby boy who was affected by X-linked HED. And what we found was giving the protein after birth is really too late to make an impact on some of the development structures to develop sweat glands and teeth. 2016, Dr. Holmschneider successfully treated 3 babies. And in Germany, they were allowed to do this, calling it a compassionate use. And his trials or his studies were extremely successful. And actually, we have 2 of these moms here with us this evening to share their story. Now, in 2017, Esper took over the development of this protein and began looking for collaborating partners. In 2018, we funded Dr. Holmschneider again to complete the NFED natural history study of X-linked HED. And in 2020, the FDA granted this therapy breakthrough therapy designation, which is really important because what that did was it expedites the process. 2020, just 3 years ago, Esper partnered with Pierre Fabre to commercially develop the therapies proteins now called ER-004. So in November of 2021, clinical trials opened in Europe for this study. And in 2002, just last year in July, the first clinical site opened in the United States. So our first clinical site in the United States is at Washington University in St. Louis, and Dr. Kathy Grange is the primary investigator for this clinical trial. Now, at this point, I'm going to hand over the presentation to Dr. Kathy Grange, who will go into a little bit more detail about EDELIFE, the clinical trials, and the science behind it. So stay tuned, and let me welcome Dr. Kathy Grange. Good evening. I'm Mary Fate. I'm the executive.
My apologies. There you go.
Okay. All right. Well, thank you for that introduction, Mary, in your absence. I'm happy to be here this evening to talk to you all about the study. And as Mary mentioned, we do have a clinical trial site at Washington University in St. Louis, and there are additional sites in Europe as well. So I'm a clinical geneticist at WashU, and I'm in the Department of Pediatrics, the Division of Genetics and Genomic Medicine. And I've been involved with the NFED since, I think, 2006, and have attended many of the meetings in the summer and participated in some of the clinical trials with Ectodermal before the studies were transferred to Rare. You want to show me the next slide, Veronica? Okay, so just a quick review. I know you all know most of this already, but X-linked hypohidrotic ectodermal dysplasia, XLHED, is a genetic condition that involves abnormal development of hair, teeth, skin, and glands, like many of the other ectodermal dysplasias. And it is caused by a mutation or pathogenic variant in a gene called EDA, or ectodysplasin A, which is located on the, excuse me, X chromosome. And I'm sorry, I gotta take a walk. So sorry. And as I'm sure you're all aware, males are most significantly affected. Women can be carriers of this condition, but can also be variably affected themselves as well, but usually more mildly than the boys and men. And EDA encodes a protein called ectodysplasin A1, which is a signaling protein that I'll explain in more detail. And we are able now to test suspected individuals of having XLHED by genetic testing on a blood sample or sometimes a buccal swab sample. And the testing is highly accurate in males. And there's many, many different types of mutations in the gene. Sometimes it's just a single letter change in the sequence of the DNA, what we call a missense mutation or nonsense mutation. And sometimes it's actually like a small missing piece or even a larger missing piece of the gene. that can cause the condition. Next slide. And this is X-linked recessive condition. So showing, I don't know if I have a pointer available. Can you guys see that? You probably can't see the pointer. So in this little cartoon here, we're showing a family here where the father does not have the condition, but the mother is a carrier. And many times women who are carriers don't have any features of it, or if they have some features, it's very mild, so they may not know they're actually a carrier for XLHED. But you can see there, the mother, because she has 2 X chromosomes, will have one X that's normal or has a working copy of the EDA gene. And then she will have an abnormal copy of the EDA gene on the other copy of her X chromosome. And every time she has a child, she passes on either one of those 2 X chromosomes. So if she passes the X chromosome with the mutation onto a daughter, the daughter would be a carrier like herself. If she passes on the normal X, she'll just have a normal daughter. And she can also pass that normal X on to a boy and have a son who does not have XLHED. And the fourth possibility is passing on the X chromosome with the mutation to a boy. And that boy will be affected with XLHED. So overall, every time a woman who is a carrier for XLHED gets pregnant, there's a 1 out of 4 chance that she will have a boy that has the condition, and a 1 out of 4 chance that she'll have a daughter that is a carrier for the condition. So next slide. And this cartoon here just shows what we call the ectodysplasia NF-kappa B signaling pathway. So I put a little box around the very top gene and protein up there, EDA, and the little dark blue cap at the top is just showing a cartoon of the ectodysplasia protein, which is the initial signaling protein in this very important pathway. And so this EDA protein initiates a whole cascade of steps in this developmental pathway that results in the embryonic and fetal development of all of the different ectodermally derived structures. So that includes teeth, hair, skin, sweat glands, and other glands of the body as well, including glands in the respiratory tract and in the eyes, and other glands of the body like mammary glands, for example. So you can see there, oops, go back for a sec for me. There's EDA is the top protein there, and below that is EDAR, which is its receptor. So that's very important for us to have a normal copy of EDA to attach to the receptor, and then the receptor, which is on the cell surface, will cause all these other genes to start producing their respective proteins, leading to a transcription of lots of different target genes that are responsible for helping the embryo and fetus develop all those important structures. So next slide. So this is just another more simple cartoon looking at what normal ectodysplasia does. So you can see it there. It's actually multiple copies of the protein stuck together, and you can see the little blue ovals there with the orange string. So that's a cartoon of the ectodysplasia EDA1 protein there, and it's attaching to the EDAR receptor on the cell surface. And then once that happens, then a whole cascade of events takes place, resulting in those structures, the teeth, the skin, the hair, and the sweat glands, and so on. So next slide. And so all of this is taking place very, very early on in embryonic and fetal development. So this is what happens if you have XLHED. Basically, you don't have a functional copy, and this is in males, because you remember females have 2 copies of the X, so they'll have 1 normal X or 1 normal copy of EDA, but boys only have 1 copy. So they don't have any, may have no functioning protein, or they may have a protein that doesn't work very well, or as well as it should. And that means that it can't attach to the receptor, and it can't result in all of those developmental steps that are supposed to take place. So go to the next slide, please. So as Mary explained, this recombinant or replacement protein was developed, and it's called FCEDA1. That's sort of the scientific name for it. Basically, what the researchers did was they attached a part of immunoglobulin molecule to the important portion of the EDA molecule, which makes it possible for that to get into the tissues where it needs to be in order to stimulate that pathway. And so this replacement molecule, as it turns out, was very successful in repairing the defects in animal models. Go to the next slide for me. So Mary mentioned some of the animal models. This is a picture of a couple different dogs that have XLHED. This is a naturally occurring animal model, and there's and as she mentioned, Dr. Casals has some of these dogs at the University of Pennsylvania that she was doing research on. And this middle panel just shows you that when this treatment was given to the dogs, and these were puppies that, this was after they were born. So this was postnatal administration of this recombinant protein. It was given to the dogs. It just shows how successful it was in correcting some of the features. So on the far left column there, you can see what the teeth, for example, look like in an untreated XLHED dog. You can see that the dog is missing a lot of its teeth, and they're abnormally shaped, very similar to what you would see in a human with this condition. In the far right panel just shows you what a normal dog's teeth was after birth. And so you can see how nearly normal the teeth are. Anyway, so this was a postnatal treatment, but this treatment does not really work for dogs prenatally because of their placenta is a little different, and it doesn't, the treatment didn't work for the prenatal treatment. Anyway, so as Mary explained, there's also a mouse model of XLHED, again, a naturally occurring mouse model, and you can see it over here on the bottom left. It's called a tabby mouse, and it has a kinked tail, and it's very small and has abnormal fur and missing teeth. So it's again quite a good model for the human XLHED condition. And in this mouse experiment done by Doctors Gaid and Schneider that Mary described to you, the pregnant mothers were given the treatment prenatally. And in this case, there was permanent correction of essentially all of the features of XLHED in the offspring. And that's shown in that bottom right-hand picture of the mouse. And you can see it had normal teeth, it had normal hair and on its tail, and without the kinking, and it had normal fur. So that was really exciting results that they were able to get from this experiment. Next slide. So then, as Mary mentioned, Dr. Schneider in Germany was able to treat 3 boys prenatally with this drug therapy. I should mention before that, we tried doing this study on boys that had already, had been born neonates, newborn boys with XLHED, and there were 10 or 11 boys that were treated postnatally, and it turned out that it was too late for human babies to be treated postnatally. It did not work. So Dr. Schneider was able to do the treatment for 3 boys on what they call a named patient basis in Germany. And then 3 more boys were treated after his initial report in the New England Journal of Medicine in 2018. And after birth, these boys have shown excellent results in terms of their sweating ability and an increase in the number of tooth germs that would be expected compared to untreated boys with XLHED. So longer term follow-up is underway to find out how things will turn out with regard to adult dentition and so on. But these preliminary studies led to the establishment of the EDELIFE trial, as Mary explained, and getting permission from the FDA and the European similar organizations in Europe to allow the studies to be done on more boys. Next slide. So this, the trial design is that it's open label. That means everybody in the trial gets the real drug. And there's only one arm, meaning everybody gets the same dose. And the genotype match controlled means that we hope to be able to find an individual within the family who is affected by XLHED but is untreated, so that we can compare within that family what the outcome would be for an untreated person with the same mutation, with the same genetic background for other features. And non-randomized means that everybody gets the drug and there's no randomization into different groups or anything like that. And the goal is to treat 20 boys, which sounds, like not very many, but it's difficult to enroll a study like this, just finding the appropriate cases to be enrolled in the study. And of course, sorry, the target population is male fetuses. So we want to enroll boys that are predicted to have classic XLHED, so that we can really tell how well the treatment is working. So only boys are being treated in this particular study, not girls that are carriers or might have milder features. And so in order to be in the study, the mother has to be a known carrier with a known variant in the EDA gene by genetic testing. And then the male fetus has to be diagnosed with XLHED. And the initial diagnosis can be done by ultrasound examination of the fetal tooth germs. So Dr. Schneider's become quite expert at this, as far as being able to look at fetuses around 16 to 20 weeks of pregnancy and count the tooth germs and compare to what would be expected at that gestational age. Another possibility is to have prenatal testing via amniocentesis or chorion villus sampling that would show that the fetus definitely is an affected male with an EDA mutation. So I should say that if the prenatal diagnosis was made only by the ultrasound testing initially, and the mom is enrolled in the study, when at the time of the first amniocentesis to give the first dose, then we do genetic testing on the fetus from a sample of the amniotic fluid at that time to be absolutely sure that it is an affected male. And then there are 3 doses of the drug, the ER004, given into the amniotic fluid. And as I mentioned, the comparator study is being conducted alongside this study for an affected male relative. Next slide. Just a little bit more about the investigational product, the ER004. It's a human immunoglobulin G1 region. So as I mentioned, it's part of an immunoglobulin molecule that is bound to the human ectodysplasia A receptor binding domain. So it's a fusion protein, basically. It's a long name, but it's the key part of the ectodysplasia molecule that's bound to a portion of an immunoglobulin molecule. And so when this is given into the amniotic fluid, what happens is the baby swallows it, because babies are constantly swallowing amniotic fluid. That's just normally what they do. And because it has this FC receptor attached to the receptor binding domain, that allows uptake into the fetal circulation, and then in the circulation in the blood, it can get to all of those developing tissues. So this is really a very unique treatment. There's never been another a drug developed that's quite like this. So that's why it's called here a first-in-class signaling protein replacement molecule. And the idea is that it will replace the missing EDA protein in these patients with XLHED, and then allow that whole cascade of events to take place so that all these structures can develop normally. But the key is that it has to be done early enough during the gestation of the baby in order to have an opportunity for those tissues to develop the teeth and the sweat glands and so on. And it's given through amniocentesis, 3 injections, approximately 3 weeks apart, beginning at 26 weeks of gestation. The next slide. And as far as the official objectives of the clinical trial, primary objective is to assess the effect of ER-004 on the sweating ability of the treated babies at 6 months of age. So when they come back for their 6-month follow-up visit, they will have a sweat test, and that will quantitate the amount of sweat that they can produce. The secondary objectives are we'll also be doing imaging of the skin to look at, actually visualize the sweat pores on the skin and count those up and see how many of them there are. Typically, boys that have severe classic XLHED basically have no sweat pores if you do that type of imaging test on the skin. I will also be assessing their dentition of their baby teeth, and also as they get older, the adult teeth. And they will be followed up to 5 years of age to look at how they do over that period of time. And also just assessing the safety of the actual intraamniotic injections, and also the long-term safety of the treatment at up to 5 years of age. Next slide. So who is eligible for this study? So for the mother, mother has to be an adult over 18 years of age with a confirmed pregnancy no later than 23 weeks, up to 23 weeks, 6 days. And again, genetically confirmed as a carrier of an EDA mutation. And then for the fetal participant, has to be a male fetus with a confirmed diagnosis of XLHED. And the untreated relative, again, in the same family, it could be a sibling or it could be an uncle, somebody in the same family that has the same EDA mutation as the treated participant. In the possibility, in the chance that there is no affected male in the family, another individual with the same mutation will try to be identified from a database and that type of thing. Next slide. And then this just outlines what the visits are like. So for the pregnant woman, the first visit is called a screening visit, and that takes place between 19 and 24 weeks. And so they come to the study site and they're evaluated to make sure that they're healthy. They have basic blood work, they have an ultrasound and some other things to make sure they're eligible for the study. And they stay on the site for up to a day and a half, something like that, if they're traveling from far away. And then once they're enrolled in the study, they have to come back for 3 more visits for each of the dose administration through the amniotic fluid. And so the first visit is around 26 weeks. Then the second dose is 28 to 29 weeks, and the third dose is 31 to 32 weeks. And for these visits, they have to stay in the hospital overnight to be observed to make sure everything's safe after they've had the amniocentesis. So they're at the study center at least a couple days. And then it is a long commitment to be involved in this study, because we are following the boys out to 5 years of age. And so there are a total of 14 visits, including the the screening visit up to the year 5. So we want to see the baby at birth if they deliver at the study hospital. If they deliver at the home hospital, that's okay. That's acceptable. But then they would come to the study center at 1 month of age for the first visit with us. The mother would also come, obviously, and would be have her follow-up visit with the OBGYN team. And then we'll see the baby back at all these different visits, 3, 6, 12, and 18 months, and then years 2, 3, 4, 5. Next slide. And this is just a little cartoon from the company, the sponsor's website, about what happens at the follow-up visits for the treated boys. And so they, of course, have a physical exam, a growth assessment. We review their medical history. They'll have EKG. They'll have some child development questionnaires. They'll have blood work, and of course, we'll check their sweating ability. We do eye examinations, looking at their teeth. And they will have 1 MRI scan to look at the teeth, and they'll have some x-rays of their teeth as they get older. The eye assessment is to look for dry eye and to look at the meibomian glands, which are the little glands inside the eyelids that moisturize the eyes. And then we'll also be doing some tests to look at how well they produce saliva. And there'll be some interviews for the mother. Next slide. And this is just a slide showing for the affected male relatives. They only come for 1 visit. So it could be, one of the visits where the treated boy is coming, the sibling or the other family member could just come along at that time. And we do similar evaluations on them. Not the MRI, but all the other things to see what their physical characteristics are like as compared to the treated individual. So I think that's my last slide. Oh, I think there's one more. So one more?
One more.
One more, sorry. Okay, so there are 7 sites open. Just me in the US so far. There's 6 sites in Europe, 2 in Germany, France, Spain, Italy, and the UK. And so far, 3 patients have been enrolled, 1 in France, 1 in Germany, and then our 1 case in the US that we've treated so far, and the baby's due next month. So I think that is my last slide.
Yes, excellent. Dr. Grange, thank you so much for all of that information. I'm sure that we will have questions as we finish up here, but we do have another some more remarks from Mary, so I'm going to play those now for everybody.
Thank you, Dr. Grange, for that excellent presentation. Thank you very much. So some of this information I know will be repetitive, but it's so much information and it's so important for you to hear and understand. So what if you want to participate? So how do you participate? So you can contact me or Dr. Grange following the webinar, and here's our emails and phone number. And if you have questions, I have an open door virtual policy. You can email me, you can call me, I'll call you right back. But surely if you have questions, please reach out because we want to make sure you have the information. And remember, timing is very important. You must join the clinical trials during the 2nd trimester, so by week 19 to 23 of pregnancy. It is very ideal if you already have a genetic test. So if you already have a test result and you have your results, that would be wonderful. So if you need help with genetic testing, please reach out to me because the NFED can help you with that. And remember, there's 3 visits during pregnancies for treatment at 26 weeks, 28, and 28 to 29, and then 31 to 32. So make sure you reach out by the second trimester so that you can get started if you're interested. And then you can see below, there's a illustration of the procedures that you will undergo during the treatment visits. You know, you have to determine if you're eligible. There'll be a medical history, a physical exam, blood test, urine test, ultrasound, and so forth and so on, OB exam, MRI scan. And then you can see here everything that is included in that visit. And then remember, you have to be prepared to follow up. There's 3 visits during pregnancy for treatment, which we talked about. And then there's visits after the baby's birth at 1 month, 3 months, 6 months, 12 months, 18 months, 2 years, 3 years, 4 years, and 5 years. And that seems like a lot, but we will make it, or Esperair will make it as convenient as they possibly can. And so that you get these follow-up visits, because it's only with this important data collected that we will be able to get this therapy commercially marketed to impact many, many other people. You know, those that participate in this clinical trial have such a unique opportunity to make a huge impact for the ectodermal dysplasia community for years to come. So just make sure you know that there are follow-up visits, and so be prepared for that. And so what's the cost of the clinical trial? So participation is free. All the costs are paid by the sponsor. Travel cost, arrangements, any other arrangements, you know, lodging, childcare, pet care. So don't let cost be an obstacle to you. If there's something that you need help with, make sure you discuss that with Esperair or me so that those arrangements can be made. So I think that's all I have for you this evening. And thank you so very much for participating. And please reach out if I can help you, or if you have any questions that we can help you with. So we want to thank our sponsors, Esperair and Pierre Fabre, because without them, this therapy would never come to market. So we are very grateful to the sponsor of this clinical trial. So again, thank you very much. Have a great evening, and reach out with any questions.
Okay, so Emily, I know you have joined us. I believe you're here. Yes, there you are. I'm seeing you now. Sorry, I was looking.
Hi.
Hi, Emily.
Hi.
So I just wanted to I turn it over to you. Sorry, Siri thinks I'm talking to her right now. So we'll just keep it moving along. But if you want to just share a little bit about, you know, your experience with the clinical trial, and I think something that's important if anybody is, you know, thinking about it, like what are maybe some of the worries that you had and how you managed that, or anything that you think. I know that you've shared with other moms, so anything that is valuable there, you know better than I. So I'll turn it over to you.
Okay, so I'll kind of go over a background. I'm sorry in advance. My kids are just in the other room and they're rambunctious. It's right before bedtime for them. We're on the Pacific time. Anyways, my name's Emily Nelson, as she stated, and my son is Finley Nelson. He is now 2 years old. We found out at 20 weeks by amniocentesis, after the results came through. It was about 20 weeks. My son showed up that he was affected with ectodermal dysplasia. At this point, we've already been in contact with Dr. Holmes Snyder, and when we found out it was positive, we wanted to proceed on treatment, and to us that was like the best thing we could, like best resource that we could try to do for our son who's affected with ectodermal dysplasia. And we did have a lot of questions. We had a lot of logistics to figure out as well, because we had to go to Germany to get the treatment. It was not available yet in the United States, and I'm so thankful it is for so many moms. So our big question was, what are the side effects? Were there any? And has anyone else had issues with it, even though there was only like 2 moms prior to me. So there wasn't really a lot of data to go by other than, previously they went and did the babies, children, adults, all those studies prior to us. But, and there was, to our knowledge, no issues, no side effects. And then our other big question was like, if our son, it was pretty 100% certain that he had it, but if there was a small chance that he did not have ectodermal dysplasia and some, with the testing didn't work out, like our concern was, would our son be okay getting the protein injections? And, it makes sense that he would be fine because these are proteins that they already have. And so those were kind of like big questions we had as a family. And so we decided to proceed. We made all the arrangements to go to Germany. It wasn't easy. It was, I had to travel alone to Germany and I had to be there for a month. I left my daughter, who was 13 months old, with my husband and my mom to watch over while I was in Germany. A lot of FaceTime, a lot of video chats. After the 1 month, I came back home for like 2 weeks. Then I went back to Germany to get the third treatment. We broke it up that way. We did the first 2 treatments. I stayed there for that. Just because it was during COVID and it was so hard to travel, hard to get through, that I wanted to make sure I got the 2 injections, and then we were going to risk it and try to go back to Germany for the third treatment, which it all worked out, thankfully. And then after all the treatments were done, I was able to go back home, be with my family, and wait for Finley to arrive. And he came at November 29th, 2020. He has skin issues, things like that, but like he's a healthy baby. After a few months, he was about 2-3 months old, I started picking him up and I started feeling his back wet. The first time I was like, okay, this could be something, it could not be. And then I started feeling it more and more often. And I also compared it with my my husband and my mom, who occasionally watched him as well. And we all agreed that like he was getting sweaty and on his back. And I think that like at that point when we realized that, it was like, it made everything worth it. All the sacrifice, everything was just like made it all worth it for him. And later on, we started noticing his head sweating and like seeing drops here down his forehead. And we went to Germany after he turned a year or close to a year with Laura. And it was great going with her, not alone this time. And with her son, we had the 4 of us. They tested his arm, both arms, to see if he had sweating on his hands by doing a test. Some kind of like electric. I don't know if it was electric or not. I'm not the scientific person. I'm not the doctor. But it started sweating, and we could see like, he has sweat cells and all those great things that we were hoping to get. So yeah, that's kind of my story. I don't know.
No, I think that's great. I think we might have questions for you, but we'll turn it over to Laura and let her share her story, which is
absolutely
similar, but also
it is.
It's and knowing about your guys' connection and traveling together is really great too. But I'll turn it over to you, Laura, and let you tell your story, and then we'll open it up because I bet people will have questions for you.
It's really good to see you, Emily. I don't think I've seen you since we went to Germany.
I know we text, but not as
it's been a long time. So I'm Laura Reiser. My husband's name is Milo. We have a 4-year-old daughter, Evie. and then a 2-year-old son, Bennett, and that's who's in the photo there. So I think I was the 5th mom to get the injections. Bennett was the 6th baby. And I've always known that I had ectodermal dysplasia, and my dad has ectodermal dysplasia. We're in Oregon, and my dad has been a part of Dr. Zanana's research up at OHSU, Oregon Health Sciences University, since probably before I was born. So it's just always something that I've been aware of, and I've always followed all of the research, read all of the articles. I was very aware of everything going on with the study, from the dogs and the mice and everything. My husband and I had made a decision pretty early on when we got married that we didn't want to have kids because we didn't want to risk passing on XLHED. But we still followed all the study, thinking that, you know, maybe one day if we ever changed our minds, it would be an opportunity to make our kids' or son's life better if it were available. So we followed it. I feel like I knew a lot about everything just from what I had read and reading the studies. And I have to say that, so Veronica asked, you know, what questions and concerns did I have, or how did I make that decision to participate? I had sought out the study. So as soon as I had found out that we were having a boy, I had contacted Mary Fate and said, you know, who can I contact? What do I need to do if we find out that this boy has XLHED? And so I had sought it out. And even so, when I got all of the paperwork and the stuff from Professor Snyder, was who I had been in contact with, he said, you know, do you want to do this? And I immediately was like, I don't know. I honestly don't know if I can do this. And I think that is okay. I think it's really hard, and it's a lot to think about. And Emily was actually, I think, one of the key reasons that I was able to go through with it. So Mary put me in contact with her, and so I'm reading all of the risks and, you know, what all of the potential bad things are, you know, that could happen to this. And Emily did 2 things that I think helped me tremendously. One of them is that she has a medical background, so she's a nurse. And although I was only the 5th mom to get these injections, what Emily explained is that the protein goes in via an amniocentesis, and amniocentesis are done a bazillion times a day all across the world. It's not a big deal, and there really aren't any additional risks with this procedure compared to amniocentesis. So it's like, okay, you know, I can put that into perspective, and I can do that. And then the other thing that she shared with me is you just have to do. So she is amazingly strong and went through so much. And that comment, I guess, you just do what you have to do for your kids. I had so focused on all of the potential bad things and, you know, the scary parts of it. And she helped me to focus on really what were the benefits that could come out of this. And once I shifted that focus, it really helped me to move forward with, you know, doing this. So I talked to her for a long time, and then I got off the phone and talked to my husband, and I still didn't know. I was like, I just really don't know if I can do this. It's too much, I think. And my husband looked at me and he said, isn't this what we've been waiting for? So I think I was waiting for it to get to the point where I could go to the doctor and, you know, just be given a prescription or whatever, not necessarily go through a whole experimental trial, but that's just not where we're at right now. And it will take, I think, all of us to do this so that it gets to that point. And so once I changed that focus and really said, okay, I'm going to do this, 3 weeks later, I was in Germany getting my first injection. And honestly, the procedure itself is not, it's so anticlimactic. It's kind of like, I don't know, looking back now, it's just ridiculous that I was so stressed out over it, but it really is, it's not more than a minute. I think I might have to rewatch the video, but I think the whole procedure, each injection is like a minute long. So it was, yeah, it ended up being totally fine. And I stressed a lot, but it was okay. And then the benefits of treatment. So I think that's what I also had to focus on in order to move forward. And I don't think that I realized what those benefits were really until I went to the family conference in St. Louis just this last summer. So myself, Bennett, and my dad went, and I've had seen my dad growing up have issues with sweating and overheating. He's very good at pushing his limits or doesn't know his limits. I don't know, but he goes all out. And so would frequently be sitting in front of the AC unit or, with a wet t-shirt. And at the conference, I realized that there's a difference between an adult overheating and a 1-year-old or a 2-year-old overheating. Those parents, they make it work. They have to. We do what we have to do. And they carry around the ice packs and the coolers and the water. But honestly, it's not even something that we have had to think about. It's just not even. Bennett can sweat completely normally. We're outside all the time. Even my kids' daycare and schools, there's mandatory classroom outside time every day, and it's just you honestly wouldn't know. He has absolutely no symptoms or issues with ectodermal dysplasia except for his teeth, which are he's got the 2 conical shaped teeth in the front, but otherwise he has no issues connected to ED at all. So anyway, I would do it all over again without hesitation. It was completely worth it, and it changed our lives completely than what it could have been.
Thank you, Laura. Thank you both so much for sharing your stories with everybody. And I want to just say, Dr. Grange, if there are questions, probably, I don't know who they'll be directed to, but we'll open it up. So if any of you all who are on have any questions, feel free to unmute, or if you want to type them in the chat, I can also share, I can read them, whatever everybody is most comfortable with. I did have a question, and I'm wondering, Dr. Grange, if you could address this. We talked about it a little bit in your presentation, but I'm wondering if you could talk more about why because of the clinical trial and the I guess the scientific needs, why the trial is only for boys, and what the implementations are if the treatment is able to go to market for girls who could potentially have XLHED as well.
Yeah, sure. Well, as I mentioned, typically girls are much more mildly affected than boys, and we can't predict based on a DNA test, for example, if it was done in utero. If it was an affected girl, we can't predict how much that girl would be affected by XLHED. Many girls and women have some decreased sweating, but it can be patchy, and they can have some decrease in their hair and some missing teeth and things like that, but it's highly variable. And I think for this initial trial, it's so important for them to enroll just individuals that have classic XLHED. Males that we know without the treatment, they would probably not sweat at all and have almost no sweat glands. So I think that's why it has to be limited right now to just a very specific population of patients. Now, I don't know what would happen in the future. Maybe there would be an opportunity for females to be treated in the future, but right now that's not possible.
So if I'm hearing you and I'm understanding it correctly, it's because you have to be able to compare it to what like the fully affected symptoms are to know
Right. And they're
how
targeting,
well it's working.
Right. And also targeting those individuals that would benefit the most from being able to sweat even a little bit more than what they would have normally. I think part of it is that it's it can be a life-threatening condition in affected males. in infancy due to hyperthermia, and also the infection, increased risk of infections, respiratory illnesses, and things like that. The boys are much more risk for those more serious or life-threatening side effects. So I think that's why right now we have to focus on that population so that we can prove that the treatment is helping those things, those clinical problems.
That makes sense. Thank you. On the slide up here, I have contact information for both Mary and for Dr. Grange. I also have a couple of URLs that have more information about the trials. So one is on our site, and the EDELIFE clinical trial is actually a site by the sponsors. So it has a lot of information and a lot of like the graphics you saw tonight about timing and everything. So if you want to reference any of that, that's a really great resource to see those. And I just want to thank everybody for joining us tonight. Hopefully, you know, this helps with understanding of the clinical trial, whether or not you or somebody in your family may be eligible to participate. And again, all the contact information is here, but you can always just reach out to us at the NFED if you have any questions, and we can connect you with the right people. If nobody has anything else, I'm going to let everybody go. So kiddos can be put to bed and all of that good stuff. So thank you all. Thank you to Dr. Grange and Laura and Emily for participating and sharing with us tonight. And thank you all for joining us. I hope you have a wonderful evening.